Welcome, and thank you for joining us for the second seminar in our Fall 2023 Science in the News Seminar series. Typically, the talks in this series have a three-part format, and after each part, we will pause for questions from the audience. If you're joining us on our YouTube live stream, welcome, and if you have any questions at any point during the talk, feel free to type those into the live chat, and we will relay them to the speaker at the end of the sections. So, tonight's speaker is Yash Rana, a fourth-year PhD student in applied physics here at Harvard, and he will be telling us about, can materials build themselves? The miraculous science of self-organization.
Thank you, Maggie. Thank you, Maya and Joe, for joining us in person. In some sense, I'm giving this talk to the two of you, and I would not have it any other way, because you guys are really important to me. Thank you. Cool. So the question that we have at hand with us today is, can materials build themselves? And hopefully by the end of this talk, you believe that the answer to this question is yes. And the process by which this happens is known as self-organization, which is the subject of my PhD, and also something I'm broadly interested in. So before we begin, I want all of us to think of a material inside this room, something that was engineered, something that was built, anything that comes to your mind in this room. I'll give you 30 seconds to do the same. Anyone want to share what they thought of? Yes, go ahead.
The foam in these chairs that we're sitting on.
Ooh, that's a good example. So the answer, the suggestion was the foam that's inside these chairs that we're sitting on. And so what I'm going to try to do is I'm going to try to put this material or any other material that you might have thought of under a particular framework. And that's what I would be calling as a top-down construction. So when we think of something that was built in this room or built by an engineer, it usually comes from the point of view of having a function in mind. For example, with the foam, it's something that needs to provide cushioning or softness for the seats we're in. And once we have this idea that, oh, I need to build something that should have this function, then you think about, oh, it needs to have a particular design, and then you think of what are the materials what are the building blocks you can use to build this, and you design the interactions in such a way that it fulfills your function. And this could be true for the foam in our chairs, it could be for the chair itself, it could be for the computer, it could be for the projector, it could be for anything. But even though all of us would have thought of different materials, one material that I'm pretty sure most of us would have overlooked, and there's a bunch of those inside this room, 6 in particular, it's us as biological organisms, we ourselves as materials. But the key difference is, unlike the foam in the chair, we're built very differently. So what I'm showing you right here is a video of early embryo development. And so development is the process by which a single cell turns into a fully formed organism. An embryo is what we call the first few stages of this process of development. And so what we're seeing here is, at the beginning of this video, you have a single cell, and then it divides into 2, and then 4, 8, so on and so forth. And it undergoes these processes. So it starts from a single blob, essentially a sphere, and over time, it creates more structures. So what I'm going to show you is a late stage embryo, not of humans, but for different organisms. But the essential idea is the same. As you go forward, you have more cells, but now they're not stationary, and they begin to fold and move to create the structures that will be important for the human or for this organism. And so the essential idea is that you start from a single cell, you divide, and even though you're spherical in the beginning, you create these structures from within, which will serve different functions. And so all of us, irrespective of where we come from, what life form we are on Earth, we started from a single cell, and so a group of cells could become a fly, a group of cells could become a bird, a group of cells could become a human. And the idea is that this is extremely different from how anything else is engineered, because unlike this top-down construction where you have the function first, here you have the building blocks that are set up by nature. Molecules, water, proteins, all these are just floating around, and the interactions between them are set by the laws of physics and chemistry. And these interact collectively to give rise to higher structures, and the function comes out of these interactions. So unlike engineered substance materials where the function is the first thing that comes up, here the laws of physics and biology set down the groundwork, and these interactions give rise to emergent phenomena that one can't predict most of the times. And so this is the essential idea of self-organization, which is the autonomous assembly of components into collective patterns that display emergent functions. And this is the only definition I'm going to share with you today, which is the idea that you have components which interact with each other to give rise to higher structure, which is fundamentally different way of construction than anything that we see in our daily lives. Cool. So with that, I'm going to share what my outline will be for today. I'm going to talk about self-organization in everyday life, just so to get ourselves oriented and to get us to realize that this phenomena occurs every day. And this, I'm going to give you an example that's at the molecular level, at the scale of nanometers, which is 1,000 times the thickness of our hair. And then I'm going to talk about how embryos know where to grow, essentially answering the question, how does a spherical blob know how to make different shapes in its body to give rise to different segments of its organism, and that's at the cellular level. And third, I'm going to talk about how whole organisms collectively organize at ecological scales, and this is at the scale of meters and sometimes hundreds of meters. Cool. And all throughout, I'll be coming back to this framework of scientific inquiry that I'm going to build for you right now, because for me, it's very important that all of you, well, you guys are scientists, so I don't need to tell anything to you guys, but if it was a more broad audience, to give the picture of how scientists think about these questions. And so this is the idea of combining experiments, which is observation or manipulation of nature, combining it with theory. And what I mean by theory is mathematical formulations that give a generalized explanation for what we see. And really the last component of this, which is essential for how we're doing science in the past 20 to 50 years, is to combine this with simulations. And what I mean by that is once you have an observation and you have some sort of generalized explanation for it, you have to really confirm if these insights are enough to explain the experimental observation that you're trying to see. So what you can do in simulations or in sort of an in silico setting is to pick up only those insights that you think are important and create an artificial world. And if in that artificial world you see the same phenomena that you were interested in, then you can be confident that you have gained the right insights from your experiments or your observations. So this is the insight I'm going to keep coming back to as I go on. So, as I said, we need to have experiments and observations. So I'm going to start with one to talk about self-organization in everyday life. So here I have something, and okay, that was actually maybe not. Let's do it again. That was way better. Cool. So soap bubbles, something we see every day, are a good example to start thinking about self-organization. And so we have these spherical bubbles that pop out, that float in the sky, but what's going deep down at the scale of molecules? So soap is a very interesting molecule because it has these two different components. It has one part which loves to be in water, and has a second tail part which loves to be in oil. And this actually explains how it works as well. So if you have a dirt or oily substance, the parts that love oil accumulate around the dirt, and the parts that love water accumulate on the outside. And so what happens is you create this spherical structure that's able to pick up dirt and take it away. So what happened here? We had these molecules. No one told them that they have to accumulate in spheres. But just because of the way the molecule was constructed, it organizes itself in these spherical structures, which helps soap do its cleaning action. So here we have an example where the molecular interactions are set up, and that gives rise to how this emergent self-organization happens, and that has a function of cleaning dirt. And so what's happening inside soap bubbles is the following. You have a thin layer of water, and all the water-loving heads accumulate at the water interface on both sides, and the oil-loving parts stick out. And this is able to stabilize the soap bubble. So because of the organization of soap molecules on this sphere, you get a stabilized bubble which can float in air and sustain itself. Another way these molecules can organize, or molecules like these can organize if they're in water, is that the oil-loving tails can come and interact with each other, and the water-loving parts can then be happy because they're now in contact with water. And so if a bunch of these ensembles come together, you can create these structures where the water-loving part are happy because they're in contact with water, and the oil-loving part are happy because they're in contact with each other, and you get something that vaguely resembles a cell membrane. And this is very similar to what actually happens in biological membranes as well. And so what we're seeing here is because of the molecular interactions, these molecules which have this bivalent structure where one part likes water, one part likes oil, you get these emergent structures which exist widespread in biology and nature. And so we can begin to understand how things so important to our cells that they provide a protective boundary come together by interactions that are set up at the molecular scale. Cool. So with that, this is 15 minutes, so I'm going to stop with this section. Just want to recap. Say that self-organization occurs all around us. Soap molecules is one example, but if you see the ripples if you see the ripples in a desert, if you see structure of trees and how they branch out, self-organization is happening all around us. The interactions between the constituents set up how these different constituents interact, and that directs emergent structures that come out. And so with the example of soap molecules, the building blocks were the molecules. They were interacting based on whether they like water or hate water or like oil. That gave rise to these spherical shells. And for example, in the case of cells, it provides a protective boundary. So we have this bottom-up structure that came up with this example of soap, and this is something that happens all around us. And so with that, I'll take questions for this part of the talk.
We have microphones, so it's a little bit easier to hear on the live stream. And as a reminder, if you're joining us on the live stream, feel free to type any questions you have into the chat, and we will relay them to our speaker. Do you have any questions? Sure, I have a question.
Yes, ma'am.
Whenever the soap molecules form these like little membranes inside of water, does anybody study those, like in analogy to cell membranes?
That's a good question. So when I say that soap molecules, so if you have soap in water, it will actually not form those structures because if you have, for example, here you have a tank of water, all the soap molecules will just come to the surface with their water-loving parts inside and their oil-loving parts sticking out, because that's the most energetically favorable structure. What the point I was trying to drive was that things like cell membranes have molecules which are very similar, and so the structures that we see inside cells vaguely resemble these soap molecules, and so we can begin to understand how cells work by thinking about interactions between these two co-block type molecules which have one part that interacts one way and the other way. Thank you.
Oh, thanks. Any other questions? All right.
Okay.
Cool. Yeah, we can either take a few moments as a break, or we can move on to the second part, as you wish.
If you'd like to keep going, so you're welcome to. We want to take a moment to get a drink of water.
I'm actually not that bad, so I can keep on going. Yeah, we Yeah. Okay. So, and no questions from the live stream? Cool. Okay. Cool. So, break. That's all you get. Okay. So, with that, let's start the second part of this talk, where I take up the complexity and I take up the length scales involved, and I'm gonna try to answer the question of how an embryo knows where to grow. So this question is inspired by this video. What I'm showing you here is a developing fruit fly. Here you have each of these small polygons is a cell, and what you're seeing is that you have this bunch of cells arranged in a football shape, and at some point, I'm going to play this again. So you have this football-shaped embryo which has all these different cells, and at some point suddenly different cells in different parts of this body start moving and start folding and moving around at the scale of tens and hundreds of cells, and you give rise to these structures that will eventually give rise to different parts of its body, the wings, the eyes, etc., etc. So how does this seemingly symmetrical object know which part of itself has to grow, which part has to retract, which part has to fold? There's very complicated mechanics that's going on there. But how does an embryo know which part of it has to grow? Because there's no external information. This is something that happens outside of the mother fly. So this is something that's happening on its own. Cool. And so we have this observation, and we're going to try to see if we can build theory and simulations around this. So what essentially needs to happen for something like this to grow is that information and material have to transport to different parts of this growing embryo. And so what people began to think was, okay, how do we know, are there examples or cases where we know that information and material do transfer in space and time? So this is something that everyone sees. And so it is actually kind of automatic. So let's just see what's happening as I drop this ink in this tank of water. So I released it at some point in this tank, and it starts spreading, it starts moving out. Material and information are now spreading. Different parts of this tank will know where they are based on whether they have ink or not, or how. And so we can begin to start to understand that maybe information can be transported by the presence or absence of material. So let's start building upon this idea of things spreading out, things diffusing out. And so the essential idea is that of diffusion is that things spread out. If I had collected all the molecules of air in this room and put them in the corner, over time they will spread out. If I had asked all of you to come and collect in one corner and let you chat, over time you will spread out. So diffusion is a fundamental process, and it's in a way in which material transports, and hopefully we can use that to transfer information as well. So let's build this. Let's take this idea of things spreading out, and let's try to build a theory that can explain potentially how embryos grow as well. And so if we have the essential idea of things spreading out, we can add some maths to it, and we get something known as the diffusion equation. So the equation does not matter. What's important is that the value of having theory in this case, or in any case, is that this equation not only explains how ink diffuses out, but it explains how anything and everything moves and spread out. So that's the power of having theory behind your observations, because you have this generalized understanding of what's happening. Cool. So next, what we'll do is we're going to use this theory to build simulations, and hopefully use that as a playing ground to build different ways in which information and material can be transported. So what I'm going to do, the first thing is I'm going to put this equation that I put down for you onto a computer and see if it reproduces the thing that we observe in front of us. So here what I have is my digital version of this vessel, and what I've done is I've kept or dropped ink at the center, and I've drawn this line, and what I'm plotting for you on the right is the concentration of this dye, or how dark the color is as a function as I'm moving across this line. So at this point, there's no dye at the beginning, so that's why you see nothing. There's a bunch of dye in the center, that's this inner circle, and there's no dye at the end. So what happens if I tell my computer to follow the equation that I've given it, and the equation basically tells over time how does the concentration at different points in space and time varies. So what we see is something very similar to what happened in front of us. We had material at some corner or in some localized space, and that spread out. But we've lost something. At the beginning, we had information that was in the center, and if I had asked different parts of this beaker, oh, can you tell me where you are based on whether you have ink or not, it would have been able to tell me something. But now all the dye is diffused out, and so basically we've lost all the spatial information that was encoded based on where the dye was. So since things diffuse out, we lose spatial information, and so something is missing. This is not sufficient to explain how different parts of an embryo might know where they are. And so what we're going to do is we're going to add something that we know that biology is capable of, and that is chemical reactions. So I'm going to let you think about a chemical reaction for a moment, and then we're going to see if we can put all of our reactions that we thought of in some generalized framework and then build upon it. So I hope all of you have thought of something. The thing that I thought of was the following. So I had my bike sitting out in the snow, and there was iron in the bike, in the chain, there was air, which had oxygen, and over time, it got rusted. So this is an example of a chemical reaction. A chemical reaction is anything where a substance changes form, or shape, or color, and so whatever you might have thought of, maybe cooking, photosynthesis, combustion, all of these are examples where molecules change from one form to the other. And so we can abstract out all the reactions that we thought of to this simple form where we have molecules of one type turning into another. There's yet a simpler type of reaction that I can think of, which is one molecule making more of itself. And this is something that happens all around us as well. Each of our cells is dividing, for example, if you think of the cells in a scale, one cell will divide into two over the scale of say, less than a day. And so the DNA in that cell becomes twice. So molecules can make more of themselves. And so what I'm going to do is I'm going to combine this idea of molecules making more of themselves and diffusion and see where it takes me. And this is exactly what Alan Turing did in the 1950s in this research article, where as a mathematician he wanted to combine the ideas of diffusion and reaction and use it as an explanation for how biology, how things work in development. And if you're interested, there's a pop-sci movie about Alan Turing called The Imitation Game, which also discusses his role in cracking the enigma during World War II, helping the Allies. Cool. So now let's add some complexity to the simulation that we saw in the beginning. So here, what I'm showing you again is a glass beaker. I showed you ink in the center, but now there's one key difference. In this inner circle, molecules will be making more of themselves. So even though molecules can spread out over time, inside this inner circle, molecules will be making more of themselves as well. So let's see what that leads us. So now what's happening is that even though things are diffusing out, they're also being generated in the center. So over time, you see this stable pattern that comes out. And so now, if that was something that was happening in this beaker, maybe because if I was continuously adding more ink, if I had asked this beaker, oh, could you tell me, if I asked different parts of this beaker where they were with respect to the center or with respect to the sides, they could tell me where they were based on reading what concentrations were at that site. And so now we are able to encode spatial information because we have diffusion, but we also have reactions that compensates for that, and we can generate steady state patterns. What I can do is I can change the location of the source of the reaction, and so now because the source has changed, the steady state pattern has changed, and based on that, you have encoded different or additional information. And so that's one way of adding spatial information in systems which just have chemical reactions and diffusion. Cool. We can add more complexity by adding different types of molecules. So if I have two sources here, green and red, both being generated in these smaller circles and diffusing out, we have more information because now different location of the cells can read not only the green molecules but can also read the pink molecules, and they can use these two pieces of information to know where they are with respect to other parts of the cell. Cool. So by having multiple reaction sources, you can encode additional information. But there's one thing we're still missing. That is the idea of time, because the patterns that we formed were steady state. So if we have something that's developing over time, things are changing, how do we encode this temporal information? And so essentially we need a clock. And so what I'm going to do is I'm going to use the same system where I had two types of molecules reacting and diffusing, and I'm going to construct a clock. And this is exactly the question that Alan Turing also wanted to answer in his paper. And so he created a reaction system which he called the oscillator. And the way it works is pretty simple. We have two types of molecules. Molecule B likes making more of itself, just like we talked about molecules making more of themselves. Molecule B also likes to make more of molecule A. So it's sort of a very optimistic, positive person, promotes itself, also promotes others. But A is what you would call a toxic person. So it inhibits molecule B, but it also inhibits itself. And so this sort of very chaotic dynamics gives rise to something very interesting. And so what you can imagine that happening here is that molecule B makes more of itself, it makes more of A, but as you make more of A, it kills B, but it also kills itself, and then over time B will make more of itself, and the cycle continues. So if you encode this sort of reaction system in your simulations, you have a repetitive pattern, a pattern repetitive in space but also repetitive in time. So what we've done essentially is encoded a clock, and these kinds of clocks exist widespread in biology, and it helps different organisms keep time as they go about. Cool. So now we've shown that if you have multiple sources, by having these interesting interactions between them, we can encode not only spatial information but also temporal information. And now I want to show you data from experiments where people have used these ideas to explain how fruit fly embryos grow. So in a fruit fly embryo, the video that I showed you in the beginning, what happens is the mother deposits one kind of protein called bicoid, the name doesn't matter, but it's deposited at its head. And so we know that things diffuse, so that gives rise to this spatial gradient of bicoid. And here, the more the red, the higher the concentration of bicoid. So we see the spatial gradient of this protein. This protein then interacts with other proteins, for example, Kr, and because of the way things react and diffuse out, this protein is only localized in the central, this belt region of this growing embryo. So there's a cascade of interactions that happen because there's not one protein, there are thousands of proteins inside these cells, and eventually you get this sort of striped pattern that comes out because of these different interactions that are happening. And once this sort of pattern is set up, that tells the different segment, that tells the embryo, oh, I have these many segments, and different segments will start doing different things, start moving, and that tells the embryo which parts should do what, and that gives rise to this spatially inhomogeneous mechanics, and that gives rise to different shapes that come out. So reaction diffusion lays out a geographical map across the embryo, and then mechanics takes over, and that gives rise to the different shapes, and eventually the whole organism comes out. Cool. So to recap this part about of embryo development, the ideas that I shared today was diffusion is the process by which things spread out. Reaction is the process by which molecules molecules can make more molecules, and if you combine reactions and diffusion, you can encode spatial and temporal information, which is crucial for embryo growth. And so here, coming back to this framework of self-organization, like bottom-up construction, the building block were these diffusing molecules. They had interactions which were set up by the reactions that we define or defined by nature, and this gives rise to spatial patterns, and that eventually leads to growth. So we have emergent bottom-up function that comes out based on the reactions that are set up at the level of the constituents. And so this is also a very good example where we start with experimental observations, we build theories for it, and then we use computer simulations to gain more insight and hopefully learn something more about the experiments that we're seeing. So with that, I'll take questions for this part of the talk.
Yeah, this is very fascinating actually. Thanks, yeah. I have a couple questions. So in the image you showed us for the float slide, there was this initial deposit of protein.
Yes.
And that kind of like was the initial condition that determined the, right?
Yes.
So like if we have like just I guess like a single cell, like initially the cell is like it's just one and then it divides. Like where does this kind of like in this division of like when the cell is growing and dividing, what like does something like let's say for humans does something like that happen where like the interaction with like the mother's uterus or something like deposits protein in a specific part of the cell and that kind of.
That's a really good question. Just to phrase it in a slightly different way, so the question is, here we have this sort of case where we have maternal deposit that sets up the initial condition for development. What happens if you have only a spherical shell, and where does the initial conditions come out of? So a lot of interesting things happen. I'll give you a very interesting example. For example, in mammalian development or the growth of humans or mice, the location where the sperm enters the egg sets up initial conditions as well. So you have a spherical egg, and then you have the sperm that comes in, and where the sperm comes in sets up the condition and the asymmetry. And for example, you have the head-tail asymmetry. I might be misstating different organisms, but at least in some organisms, depending on where the sperm comes in, that sets up the initial point, the initial asymmetry, and then things follow. Yeah.
Maybe as a follow-up. So like I don't know, maybe this is not known, or maybe I don't know. Is it believed that like for instance for humans, once the egg is like fertilized, that on its own can develop into an embryo, or is there like does it need like maternal input consistently during the growth?
So I think for human growth, you do not need any maternal input until the first 8 days of growth. So the first 8 days of growth can happen outside the mother as well. And that's what happens in IVF clinics. But after that, there is input, at least of like nutrients that comes from the mother. How much that sets up spatial gradients, I don't know, but you can imagine that since this input is spatially homogeneous, I would imagine that would be some information as well. Yeah.
Very interesting.
Do you have any other questions from the audience?
Yeah, I'm also curious about the sort of irreversibility at some point. So when you set up this gradient, basically, so the protein comes in, is it at that point that, let's say, for some reason, the protein were also to come in from the other side, have all of the cells already decided at this point, like, we got this input, now we're sort of cascading down this fate, and especially when they're moving in the organism, and you'd set up those bands earlier, and if you move from one band to the other, at that point you've decided what you're going to be, but when is that time point? Do we kind of know about that?
That's a good question. And so the question is that you have these different stripes that are come out, and at what time does these two different parts decide who they are? So I think so. I'm not sure in the case of fruit fly embryos when that happens, but there are for example, I know for example for human embryos at least, I think the first sort of idea of cells being different happens at the stage of 8 cells. But I guess what I'm saying is, for this particular case, I do not know when they start, but I do know that you have to have the pattern formed first before different cells start deciding what they do. So in some sense here, they might not be differentiated by means of them deciding what type of body part they'll be, but they are differentiated based on the phenotype that they start displaying at the stage. So they differentiate in the sense that they start displaying different mechanics. Yes, but are they decided that I am going to be a hair, or I'm going to be the foot or the head? That I'm not certain. Yeah. Good questions. Yeah.
Do we have any other questions? All right, as another reminder, if you're on the live stream on YouTube, feel free to type any questions you have in the chat. Yeah, great. Oh,
Yeah, maybe one more question. So in the model you showed us of the clock, like this clock was like oscillatory, so like, but presumably the embryo growth is like it's not like it grows in day and shrinks at night, right? It's like it keeps going on, right? Is the model of the clock in the case of like a growing embryo like just keeps continuing, or is it also oscillatory?
So in the case of this embryo growing, I think I would. I think the idea for that simulation was to show that you can have these clocks, and eventually, once the organism is fully formed, there will be clocks, for example, to tell the organism that, oh, now it's daytime, now it's nighttime, based on different inputs. I think at this scale, there is no sort of clock in the sense of things going back and forth. And in fact, going back to what we were talking, there is this irreversibility where once things are decided, they go only in one direction. So at this stage, there is no clock in that sense. Yeah, yeah.
great. If we have no more questions, we can move on to the third part.
Cool. Yeah, if you guys want to take a break, that's also fine. But if not, we can also go to the next part. Okay, cool. So the next part of this talk is going to talk about self-organization at a very different scale, at the scale of whole organisms, in the context of ecological systems like flocks and collective organizations of animals. So flocking birds is something that we see all around us. It's very aesthetically pleasing to see these, and the more interesting part about it is that there are no leaders in this organized system. There's no leader bird telling each other bird where to go, and there's no external information. So we get these sort of floating collectives of organisms with no leaders. So these structures emerge bottom up from the the birds itself, and these sort of organizations exist all across nature. For example, for example, the schooling of fish, you have these large tornado-like organizations where the size of these ensembles are hundreds or if not thousands times bigger than the size of the individual. So you have information being transported at the scale thousands of times larger than the scale of the individual. So how do these structures come out and how do they behave is something that has attracted physicists for a very long time. Part of the reason being of the way how they respond to perturbations. So here I showed the example of a school, a fish, and what you might be able to see is that there's a seal that's trying to invade the school. And the way the school responds is not reminiscent of a single fish responding. It's sort of a collective response because you see the whole school clears out. So this whole thing is responding as if it was one coherent material and not a collection of fish, not a collection of individuals. So you have these properties that emerge out, and some people would like to compare this as a drop of liquid or a drop of a gel. And so physicists have been really attracted by this idea of collectives, of animals, and how do you describe them, and how do you describe the fact that they organize in this way. And so the first and so here what we're going to do is we're going to start with an experimental observation, and we're going to go straight to building computer simulations of that. And that's partly historical of the way this system was studied, and partly because there's no good theory or set of equations that anyone has been able to write down that convincingly explains or is able to give an understanding of finite-sized flocks in the sky or in the ocean. Cool. So the first physicist that thought about these systems was this guy called Vishek, and he saw that, oh, I have these sort of objects that are floating in the sky, and it seems that they want to go in the same direction, and that reminded him of magnets. And physicists are very notorious for bringing out things like springs and magnets to whatever they see. So what Vishek knew is that if you have a magnetic substance, the internal orientation of these atoms tend to align at low temperatures. And he was like, oh, I'm going to use this essential idea and I'm going to impose it on biology and see if it works. That was the idea. And so he came up with this algorithm or model for how the flocks might be working. And his idea was that, oh, I'm going to take all of these birds as pointy vectors or pointy magnets swimming or flying in the sky. I'm going to impose a rule that each flocker or each bird looks at a radius r around itself, and it bases its velocity based on the average velocity of its neighbors. So this sort of local interaction was something that he came up with, and he was like, oh, is this sufficient to see this emergent flocking behavior? And so that's what he did. The key idea was that birds are like magnets, they tend to align, and then he formalized this by giving a set of rules for all the birds that were flocking. So the simple rules were that birds move in the direction in which they're pointing, they tend to align with their neighbors, and another step was that the decision making of this alignment is error prone, and that's something that was set up from the get-go. What do I mean by decision making being error prone? If the error is high, and if the flock on average is trying to move upward, and this bird is looking at its neighbor, if the error in decision making is high, that means the way this bird averages the velocity of its neighbors is kind of crappy, and so even though it sees its neighbors, it makes the wrong decision of where to go. And if the error is intermediate, it sees the direction of its neighbors, even though the flock might be going upwards, it makes a slight error in where it's going. And if the error in decision making is low, what this means is that as the flock is going upwards, it makes a good estimation of its neighbors and chooses that direction. And so essentially the idea was, can we use this error in decision making? Can we use the idea of these magnets flying in the sky to reproduce flocking? So what I'm going to do is I'm going to put these ideas into an algorithm, and I'm going to put this in a computer and see what it gives me. So if I do the following, where I have this error in decision making relatively high, what I see are these tiny magnets that are trying to go forward. They're looking at their neighbors, but they're making crappy decisions of where their neighbors are moving, and so this gives rise to a disorganized mess. So the question that Vishek asked, and the question I want to answer is what happens if I reduce the error in decision making? So if I reduce it to a medium, to an intermediate amount, we see that birds or these flockers are aligning to their neighbors, but there's no large-scale organization that's popping out. And so what happens if I reduce this error making even further is something remarkable, because what you have is not just local alignment, but you have this large-scale alignment where the whole flock moves in the same direction, and you also see these spontaneous changes in direction that come out. And so what we've essentially done by having these simple rules is to recreate collective transport. And so all we needed was local alignment, and that was enough to give rise to this emergent collective behavior. And so even though this is not a legit or legal representation of how birds flock, because birds are complicated, their interactions are presumably more complicated, what Vishek showed was that all you needed was local alignment, and that is sufficient to give rise to this emergent collective transport. And that's the power of taking out insights and putting them in computer models and seeing what you get out. Cool. So a quick recap on this last part of ecological self-organization is that these collectives are omnipresent in biology, and even though at first they may seem highly complicated, the only thing that's important, at least from what I showed you, is that local alignment is sufficient to give rise to this collective organization. So mind you that these interactions are happening at the scale of individuals. For example, in the case of birds, each bird is known to look only at its 7 neighbors. But if each bird is doing the same thing at all the different parts of the flock, that gives rise to an emergent collective flock. So that's incredible in my opinion. And so again, we have this bottom-up assembly, bottom-up construction that's happening here. The building blocks are the birds. They're interacting based on this alignment that was put up in the Vishek model that gives rise to collective flocks. And these flocks serve purpose, have function. For example, protection from predators, or serving as mating grounds. Yes, so with that I want to end this part of the talk and take questions. Yeah.
I guess it kind of depends on how you're doing the error for the stuff. If you're just adding noise on top, maybe this doesn't matter, but can you overcome high error with just averaging more neighbors? And is that something that people look at with sort of how flocks interact? So I know you mentioned the 7 birds, but for the simulations, does that factor into it at all?
That's a good question. And so the question is, so here I've set up this interaction where birds are interacting in some radius, and they have this where is that error? Yeah, they have this error in decision making. So the question is, can you overcome error by just averaging over more birds? So I guess the way I've done these simulations is by adding a noise. So in that sense, if I average over more but I add noise, that doesn't change anything. But in terms of actual research done on this, so the way these simulations have been done are always been by adding noise. But that's a very interesting way to think about this, particularly in the ecological context of what actually is happening, because birds are not adding noise. And one can imagine that averaging over more is better because you get more information. Yeah, but as far as I know, no simulation has looked at this particular idea. Everyone has been adding noise as error. Yeah, that's a very good question.
Yeah, because I guess you can add noise at the end for, you know, you make your average and then whatever, we have some noise term.
Yes.
But I wonder if it's almost more ecologically relevant to be like, I'm measuring your velocity with some added noise to that particular
Yes.
measurement, and then if you sort of average out or more maybe than that. But anyway, details. That's cool.
No, that's a good that's a very good idea actually. Yeah, and as far as I know, no one has done that. Yeah.
Thanks. Two clarification questions, I guess. Kind of following up on the previous question. Is this usually thought of as the bird pays attention to its X number of closest neighbors, or to every bird within a certain radius?
That's a great question. So in this case, these birds look at their radius and look at their at some radius from themselves, and they average over any bird that's inside. And so that's sort of called as I think geometric averaging. There's another way of averaging where you look at your at your x nearest neighbors, and it doesn't matter how far away they are. We just look at those 7. In fact, in birds, what was found that it's the second kind, where you look at your 7 neighbors, irrespective of where they are. And if they're closer, you just look at those 7. If they're further, you look at them, irrespective of how far they are. So that's called topological averaging, as opposed to just like geometric averaging, because you don't care where they are, you just care of like the fact that they are your neighbors. Yeah, that's a good question.
Interesting. And was that 7 number just determined empirically? Like why have we settled on 7?
That's a good question. So the way that was figured out was, first of all, there were these experiments that were done where people tracked flocks in the sky in 3D. So when all of that data came out, and in fact, you might find this surprising, that all of these things, people were doing all of these simulations for at least 10-15 years before someone was actually went out and took the data from a flock of birds. And when they did that, and what you can do is you can calculate things like correlation functions. What that means is you look at a bird and you look at what its neighbor is doing. You look at a bird and you look at what its second neighbor is doing, and so on and so forth. And so different interactions that you set up will give rise to different predictions for these correlations. So, for example, if it was topological, if it's your 7 neighbors, it doesn't matter where you are, you will see a different prediction for how neighbors should be pointing with respect to each other. So people did these experiments, and they calculated these correlations of what neighbors are doing, and they figured out that no, it's not that birds are looking at a certain distance, it's that they're looking at certain neighbors. Yes, that's a great question actually.
Thank you. Just one more. When you
Go
show
ahead.
your simulation plots, do the edges wrap around to each other?
That's a good question as well. So in these, yes, the simulations do wrap around, because when birds are exiting, in fact, like these birds see these birds that are coming from the right, and then they start going together. So they do wrap around, and so in that sense, this is sort of an infinite system. But to the point which I was making earlier, people have studied these infinite systems, and they've come up with equations that explain very well these infinite systems. But flocks are finite. We know that flocks may have only up to 100 birds. So how do we have a theory for finite-sized flocks? And that's something physicists have been trying to do very recently, and no one has a good answer.
Thank you.
So in the framework that you presented, you're like kind of differentiating between computer simulations and theory, but presumably like just the model you described, that's a theoretical model, right? Like why are you saying that we don't have?
Oh yes. So Okay.
Like this averaging over like the nearest neighbors with some error, that's like presumably some theoretical model, right?
Yes. So I guess that thing of averaging, that's sort of like an algorithmic way of explaining or like setting up a simulation. When I say theory here, I mean like sort of like a set of equations that explain that describes how a set of equations that universally describes how numbers or densities are changing over time. So in that sense, there's no equation for how densities are changing over time. That sort of simulation is sort of like a microscopic model. So it is a theoretical model in that sense, in the sense that you have a microscopic rules which are equations in that sense, but they are not based on more fundamental physics. So for example, when I write down something like the diffusion equation, it's based on more fundamental physics of things moving and having, you have fluxes because of concentration gradients. So it's based on more fundamental physics. When I'm imposing rules of alignment, there is no fundamental physics there because I'm just imposing these rules. So in that sense, it's not a theory. Yeah. So, for example, what one would like to do is to start with something like a continuity equation, and then impose things like some sort of interactions which are based on some understanding of how these things are interacting, and then an equation would emerge out. Yeah.
It looks like the simulation you presented, that looks like an easy model, like isn't like a model?
Yes.
Like equations, like
Yes.
just coupling between nearest neighbors.
Yes. Yes. So for example, the Ising model in that sense is also sort of like a sort of a coarse grain imposed interaction, and also like of course it has all of the you study the Ising model for the Ising model, but again it was constructed as a toy model, right? So in that sense, this is also a toy model for how these things work.
So you think there is like still something to be uncovered in the, like some underlying like
Yeah.
model for which this is an emergent like average system? Do you think this is something that could be found?
So I think there's something that could be found when size limits are extended to infinity, it would reproduce the same thing as these. Yes, yeah. So we lack the finite scales, we lack the finite scale effects in any theory.
Yes. One of the questions that I always like to ask all of our speakers is either what brought you to this field of research or what excites you about this research?
That's a nice question. In fact, like that kind of, that's so also takes me to what I do for my work as well. So I think for me, what really got me into all of this was things like these videos of flocks flying in the sky, because first of all they're just aesthetically pleasing, and second of all, the idea that there's no there's no leader telling this whole flock where to go. There's no master plan, there's no instruction. What is known is just that there are these local interaction, and this sort of large-scale structure emerges bottom up. It was not created for the flock to swim around like a blob of liquid. It just emerged out. No one could have predicted it. And so just this fact, embracing the chaos but giving rise to some sort of emergent structure which has some purpose, that was what inspired me to learn about all of these things.
Awesome. Great. If we have no further questions, thank you so much for being a speaker. It was a great talk.
Thank you.
And then also for everyone joining us in person and on the live stream, we'll have another seminar at the same time next week. So Wednesday, FB103 or on the live stream on YouTube. Hope you can join us.
And thank you so much for came in person. This is really, I really appreciate it.